Good afternoon, class. Today we're going to cover Unit 4, Chapter 3, Part C. This is the last chapter before your test. So now we're going to look at cells and their components. Part 2, the cytoplasm. So all cellular material that is located between the plasma membrane and the nucleus is the cytoplasm. So what's inside the cytoplasm? There's the cytosol, which is the gel-like solution, which is aqueous and has soluble molecules such as proteins, salts, sugars. There are inclusions. These are insoluble molecules, vary with cell type. For example, some cell types would have glycogen granules, some would have pigments or lipid droplets or vacuoles or crystals, and the organelles. So this is going to be our primary focus today, the organelles that make up these cells. So it's basically the metabolic machinery structures of cell, each with specialized function, either membranous or non-membranous. In other words, some of them will be enclosed by a membrane and others will not. Section 3.7, cytoplasmic organelles. So of the ones that are membranous, we have the mitochondria, the endoplasmic reticulum, the Golgi apparatus, peroxisomes, and lysosomes. Of those that are non-membranous are the ribosomes, the cytoskeleton, and the centrioles. So what's the purpose of a membrane? It allows for compartmentalization, which is crucial to cell functioning. Let's start with the mitochondria. Mitochondrion is singular, mitochondria is plural. It's the powerhouse of the cell because it produces most of the cell's energy molecules in the form of ATP, and it does it using oxygen. So it's an aerobic process called aerobic cellular respiration. So mitochondria are enclosed by double membranes. There's an inner membrane that has many folds, and these folds are called cristae. And the cristae are embedded with membrane proteins that play a role in cellular respiration. Mitochondria contain their own DNA, RNA, and ribosomes. So they're independent of the nucleus of a cell. They resemble bacteria, and they're capable of the same type of cell division bacteria use, which is called fission. So this is what a mitochondrion looks like. So notice there are two membranes, an outer membrane and an inner membrane. And the inner membrane, the little folds are called cristae. The inside, that compartment within the inner membrane is called the matrix. Figure 3.14, mitochondrion. So there's the cell, and inside you have those bean-shaped structures called the mitochondria. So if we look at one mitochondrion, you can see the outer mitochondrial membrane and the inner membrane, and the little folds are called cristae, and within that inner compartment is called the matrix. Now notice within the matrix there's mitochondrial DNA. So basically the mitochondrion has its own DNA and it's got its own ribosomes. This figure shows you how a mitochondrion divides and separates. Okay, this is very similar to the way bacteria divide, as you will see next year in your microbiology class. So first it starts off as small and it grows, and then it's going to undergo fission. So it's going to separate in two and become two mitochondria. The next organelle are known as the ribosomes. Okay, so these are non-membranous, and their job is protein synthesis. In other words, making proteins for the cell. What is a ribosome made up of? It's made up of protein and ribosomal RNA, which is different from other RNAs. So this rRNA is specific to ribosomes. It's called ribosomal RNA. So there's two switchable forms of ribosomes in the cell. There are free ribosomes and membrane-bound ribosomes. The free ribosomes, these are free-floating. So it's the site of synthesis of soluble proteins that function in the cytosol or other organelles, whereas the membrane-bound ribosomes are attached to the membrane of the endoplasmic reticulum. So it's the site of synthesis of proteins that will be incorporated into membranes or lysosomes or for exocytosis from the cell. A ribosome is made up of two subunits, a large subunit and a small subunit, as we can see in this figure. In this figure, we can see the ribosomes that are attached to the rough endoplasmic reticulum. So those little blue dots are ribosomes, and that's where protein synthesis is occurring, and that happens in the rough ER, as opposed to the smooth ER that has another function. This figure shows the two types of ribosomes, the free ribosomes that are floating free, and the ribosomes that are associated with the rough endoplasmic reticulum. Next, we're going to talk about the endoplasmic reticulum. So it consists of a series of parallel interconnected cisterns, which are flattened membranous tubes that enclose a fluid-filled interior. The endoplasmic reticulum is continuous with the outer nuclear membrane. There are two types, the rough ER and the smooth ER. This figure shows the two types of endoplasmic reticulum. Rough endoplasmic reticulum is called rough because it's studded with ribosomes, and the smooth looks like a system of interconnecting pipes. And notice the rough endoplasmic reticulum is continuous with the outer nuclear envelope. Figure 315, the endoplasmic reticulum. So here, if you look at its position with respect to the cell, okay, so the purple part there, that's the nucleus, okay, so notice how it's continuous with the nucleus and the rough endoplasmic reticulum are continuous. Figure A, you can see the cisterns of the rough endoplasmic reticulum. You can see the ribosomes on the outside, whereas if you look at the smooth endoplasmic reticulum, it's smooth in appearance. Figure B is an electron micrograph of both the smooth and rough endoplasmic reticulum. It's been magnified 25,000 times. So the rough endoplasmic reticulum it's the external surface. The external surface appears rough because we said it is studded with these attached ribosomes. It's the main site of protein synthesis that will be secreted from the cell. So it's the site of synthesis also of many plasma membrane proteins and phospholipids. Proteins will enter the cisterns as they are synthesized and are modified as they wind through the fluid-filled tubes. The final protein is enclosed in a vesicle and it's sent to the Golgi apparatus for further processing. On the other hand, the smooth endoplasmic reticulum is a network of looped tubules that are continuous with the rough ER. The enzymes found in its plasma membrane, remember they're integral proteins, they function in lipid metabolism. So breakdown of lipids, cholesterol and steroid-based hormone synthesis, the making of lipids for lipoproteins. Also for absorption, synthesis, and transport of fats, detoxification of certain chemicals like drugs, pesticides. So something like the liver would contain a lot of smooth endoplasmic reticulum because its job is to detoxify drugs and pesticides. Or for example, the testes, they produce testosterone, so the testes would have lots of smooth endoplasmic reticulum. Also, the smooth ER will convert glycogen to free glucose. So again, another function of the liver. And also the smooth endoplasmic reticulum will store and release calcium. So the sarcoplasmic reticulum is a specialized smooth ER which is found in skeletal and cardiac muscle cells. The Golgi apparatus is an organelle that it's made up of stacked and flattened membranous cistern sacs. its job is to modify, concentrate, and package proteins and the lipids that it received from the rough ER. There are 3 steps that are involved. The transport vesicles from the endoplasmic reticulum will fuse with the cis interface of the Golgi. The proteins or lipids taken inside are further modified, tagged, sorted, and packaged. And the Golgi then is like the traffic director, controlling which of the 3 pathways final products will make as new transport vesicles pinch off from the trans or outer face of the Golgi apparatus. The figure on the left shows you the Golgi apparatus on its own. So the cis face is what receives the incoming transport vesicles. And then there's packaging that occurs within the lumen of the cisternae. And then the trans face is going to make sure, will form the secretory vesicles that will send whatever was inside that was sorted to different destinations. On the right, you can see the Golgi apparatus, which is very strategically located next to the rough endoplasmic reticulum. And there you see it, with the transport vesicles. So they can either be used for exocytosis, or it can also process molecules that are coming into the cell via endocytosis. Figure 316, again, the Golgi apparatus. This one is from your book. Figure A, many vesicles in the process of pinching off from the Golgi apparatus. So notice there's the cis face, receiving side of the Golgi apparatus. Then there's a little cisterns, and then there's formation of new vesicles at the level of the trans face. That's the shipping side of the Golgi apparatus. So depending on its contents, the final transport vesicle can take one of 3 pathways. Pathway A, the secretory vesicles containing proteins to be used outside of the cell, will fuse with the plasma membrane and exocytosis contents. Pathway B, the vesicles containing lipids or transmembrane proteins, will fuse with the plasma membrane or organelle membrane, inserting contents directly into destination membrane. And pathway C, the lysosomes containing digestive enzymes remain in the cell, holding the contents in a vesicle until needed. Figure 317, processing and distribution of newly synthesized proteins. So in part 1, the vesicles move from the ER to the Golgi. The vesicles containing proteins bud off of the rough ER, they migrate and fuse with membranes of the Golgi apparatus. Then the proteins are modified. Inside the Golgi apparatus, some sugar groups are trimmed while others are added. In some cases, phosphate groups are added. Proteins are distributed. So proteins are tagged or addressed, sorted, and packaged in 3 different types of vesicles, which bud off the concave trans face, the shipping side of the Golgi stack. These vesicles have 3 possible fates depending on the proteins they carry. So in 3A, the proteins, these ones are for secretion. So proteins destined for export are packaged into secretory vesicles that migrate to the plasma membrane and dump their contents by exocytosis. In 3B, these are proteins destined for membranes, so vesicles containing lipids and transmembrane proteins destined for the plasma membrane or other cellular membranes fuse with and are incorporated into those membranes. And pathway C, these contain digestive enzymes. So inactive digestive enzymes are packaged into vesicles that eventually become lysosomes or fuse with lysosomes. Peroxisomes are membranous sacs that contain powerful detoxifying substances that neutralize toxins. So free radicals are toxic. They're highly reactive molecules that are natural byproducts of cellular metabolism. These are always being produced, and they can cause havoc to the cell if they're not detoxified. There are 2 main detoxifiers, oxidase and catalase. Oxidase uses oxygen to convert toxins to hydrogen peroxide, which is itself toxic. However, peroxisome also contains catalase, which converts hydrogen peroxide to water, which is harmless. Peroxisomes also play a role in the breakdown and the synthesis of fatty acids. Now let's look at lysosomes. These are spherical membranous bags containing digestive enzymes. They're considered safe sites because they isolate potentially harmful intracellular digestion from the rest of the cell. Their job is to digest ingested bacteria, viruses, and toxins. So something like a white blood cell would have lots of lysosomes. They also break down non-functional organelles. Also, they have metabolic functions. They break down and release glycogen, break down and release calcium from the bone. And intracellular release in injured cells causes cells to digest themselves, called autolysis. Here's a figure of a cell, and the arrow is pointing to the lysosome. Clinical homeostatic imbalance 3.3. So lysosomal storage diseases result when one or more lysosomal digestive enzymes are mutated and do not function properly. There's a disease called Tay-Sachs. It's actually a condition in which the patient lacks a lysosomal enzyme that is needed to break down glycolipids in brain cells. So the glycolipids, as a result, will build up and interfere with the proper functioning of the nervous system. It's seen predominantly in infants of Central European Jewish descent, and it causes seizures, mental retardation, blindness, and death before age 5. The endomembrane system consists of membranous organelles discussed so far. So the ER, both the rough and smooth, the Golgi apparatus, all the secretory vesicles, and lysosomes are part of the endomembrane system, as well as the nuclear and plasma membranes. These membranes and organelles work together to produce, degrade, store, and export biological molecules, and also to break down potentially harmful substances. Figure 319 shows the components of the endomembrane system. So we start with the nuclear envelope, which surrounds the nucleus, which is connected with the smooth endoplasmic reticulum. So there's all these secretory vesicles that are moving around. Then there's the contents, let's say, from the smooth and rough endoplasmic reticulum will pass through the Golgi apparatus. And then there's transport vesicles, and we've already talked about that. And also lysosomes are involved with in this endomembrane system because they're part of intracellular digestion. The cytoskeleton, cyto means cell, skeleton, so the skeleton of the cell, is an elaborate network of rods that run throughout the cytosol. Hundreds of different kinds of proteins link the rods to other cell structures. So it acts as the cell's bones, ligaments, and muscle by playing a role in movement of the cell components. There are 3 types: microfilaments, intermediate filaments, and microtubules. This figure shows the 3 components of the cytoskeleton. We can see the intermediate filaments, the microtubules, and the microfilaments, which are made up of the protein actin. So let's start looking at microfilaments. These are the thinnest of all the cytoskeletal elements. We said that they're made up of the semi-flexible strands of protein actin. Each cell has a unique arrangement of strands, although share a common terminal web, which is a dense cross-linked network of microfilaments that are attached to the cytoplasmic side of the plasma membrane, and it strengthens the cell surface and helps to resist compression. Some are also involved in cell motility, so the movement of the cell. For example, if you think about macrophages, so types of white blood cells that have pseudopods and move around. Also changes in cell shape or in the process of endocytosis and exocytosis. Figure 320a. Cytoskeletal elements support the cell and help to generate movement. So in A, we see microfilaments, which are made up of strands of a spherical protein subunits called actin. Okay, so the whole thing there is called the microfilament, and the little beads are the actin. The intermediate filaments are, as the name suggests, the size is in between microfilaments and microtubules. They're tough, insoluble, rope-like protein fibers. They're composed of tetramer fibrils twisted together, resulting in one strong fiber. They help the cell resist pulling forces. So filaments will attach to desmosome plaques and act as an internal guy wires. Some have special names. They're called neurofilaments in nerve cells. This is something we'll see next semester. And keratin filaments in epithelial cells, which we saw in the lab this week. Figure 320b, cytoskeletal elements support the cell and help to generate movement. So here we see the intermediate filaments. They're tough, insoluble protein fibers constructed like woven ropes composed of tetramer fibrils. And yes, okay. Microtubules are the largest of the cytoskeletal elements. They consist of hollow tubes composed of the protein subunits called tubulins, which are constantly being assembled and disassembled. Most will radiate from a centrosome area of the cell. So their job is to determine the overall shape of the cell and distribution of organelles. So many organelles are tethered to microtubules to keep organelles in place so they don't just float around in the cytosol. And many substances are moved throughout the cell by motor proteins, which use microtubules as tracks. This figure shows the centrosome, which is made up of two centrioles, which are made up of microtubules. 320c, cytoskeletal elements support the cell and help to generate movement. So here we see microtubules, which are made up of, they're hollow tubes of spherical protein subunits called tubulin. So we mentioned motor proteins. These complexes that function in movement, they can help in movement of organelles and other substances around the cell. And they use the microtubules as tracks to move their cargo. And they're, of course, powered by ATP. There's some actually very interesting videos out there showing how they move. You have the motor proteins would be something like dynein and kinesin, and they're moving, for example, cargo. Picture like, those vesicles, those transport vesicles, they don't just bump around and float around in the cell. They're actually moving along the tracks, and they're being moved by these motor proteins. Centrosomes and centrioles. Okay, so I showed you a picture before. The centrosome is located near the nucleus. It means cell center. It is a microtubule organizing center consisting of a granular matrix and a pair of centrioles that are barrel-shaped microtubular organelles that lie at right angles to each other. So newly assembled microtubules radiate from the centrosome to the rest of the cell. Some microtubules aid in cell division, and some will form cytoskeletal tract system. Centrioles also form the basis of cilia and flagella, which are locomotory organelles. Let me just fix that. Cilia and flagella are not organelles, they are structures. They're locomotory structures. Figure 321a, we see a centrosome, which is made up of a pair of centrioles, and these are important for organizing the microtubules. Figure B, in figure 321b, we can see this is an electron micrograph of a cross-section of a centriole. You can see that there are 9 sets of 3 microtubules. So we call this a 9-3 arrangement of microtubules. Section 3.8, cellular extensions. So certain cells have structures extending from the cell surface. And examples would be cilia and flagella, which help in the movement of the cell or of materials across the surface of the cell. And microvilli, These are finger-like projections that extend from the surface of the cell to increase the surface area, usually for let's say, for digestion or absorption or secretion. Figure on the left shows cilia in the walls of the bronchi, and on the right, the figure shows the flagellated sperm. This figure shows a section of the small intestine, so you can see the villi, and if you look at the small section of the villi, if you magnify it, you can see that there are microvilli for increased digestion and absorption of nutrients. Let's look at cilia and flagella a little more closely. So cilia are whip-like motile extensions on surfaces of certain cells, such as the respiratory cells of the trachea, for example. So thousands of cilia work together in a sweeping motion to move substances like mucus across the cell surface in one direction. And for respiratory cells, it would be towards the direction of the mouth, because we don't want mucus and debris to accumulate in the lungs. Flagella, on the other hand, are longer extensions that propel the whole cell. So an example would be the tail of the sperm. So both structures are made up of microtubules that are synthesized by centrioles that are called basal bodies because they form the base of each cilium and flagellum. This figure shows the ultrastructure of the cilia and flagella. So if you notice on the left, the basal body, also known as the kinetosome, is what gives rise to either cilia or flagella. They're both basically, cilia are shorter and flagella are longer, and there's usually more cilia than flagella in a cell. So if you follow it to its apical surface, to the outside, you will notice that both cilia and flagella have a 9-2 arrangement of microtubules. So cilia and flagella have a 9-2 arrangement of microtubules. So there's basically 9 sets of double tubes surrounding a central pair of doublets, which is slightly different from the 9-0 pattern of centrioles. And it's interesting because in some books, they call it a 9+0 pattern, because there are no tubules in the center, but in other books it's often referred to as a 9-3 arrangement, but it's just a detail. So cilia movements alternate between a power stroke and a recovery stroke. This alteration produces a current at the cell surface that moves the substances forward. This is figure 322, the structure of a cilium. A cilium is the singular for cilia. So again, note the 9+2 arrangement of microtubules. There are 9 sets of 2. Figure 3.23, ciliary function. So in figure A, it just shows you the movement of cilia. So there's a power or propulsive stroke and then a recovery stroke. It's pretty much similar to when you row a boat. So in figure B, traveling wave created by the activity of many cilia acting together propels mucus across cell surfaces. So you can see that layer of mucus that's being moved in one direction. Microvilli. Okay, so these are minute finger-like extensions of the plasma membrane that project from the surface of select cells, for example, the intestinal cells that I've shown you a picture before, and also kidney tubule cells. Certain kidney tubule cells will have microvilli, which we'll cover next semester. And the purpose is to increase the surface area for absorption. So they have a core of actin microfilaments that is used for the stiffening of projections. 3.24, we can see an epithelial cell that is made up of microvilli. So you can see at the figure on the right that at the base you would have actin filaments and the terminal web that we mentioned earlier. Part 3 of this PowerPoint is the nucleus. It's the largest organelle, and it contains the genetic library of blueprints for the synthesis of nearly all cellular proteins. So it basically contains the instructions to make those proteins. It responds to signals that dictate the kinds and amounts of proteins that need to be synthesized. Most cells are uninucleate, which means they only have one nucleus. But skeletal muscle, certain bone cells, and some liver cells are multinucleate. That means they have many nuclei. And red blood cells are anucleate, means that they don't have a nucleus. Section 3.9, structure of the nucleus. So these are the structures that are associated with the nucleus, the nuclear envelope, the nucleoli, and chromatin. 3.25a, the nucleus. So you can see that kernel, that round structure, which is made up of a nuclear envelope studded with pores or nuclear pores. And inside you have chromatin, which we'll talk about. And the innermost is called the nucleolus. Let's start with the nuclear envelope. So it's a double membrane barrier that encloses the jelly-like fluid called the nucleoplasm. Okay, so the nuclear envelope is in fact two membranes. So the outer layer is continuous with the rough ER, and like the rough ER, is studded with ribosomes. Whereas the inner layer, called the nuclear lamina, is a network mesh of proteins that maintains the nuclear shape and acts as scaffolding for the DNA. We have nuclear pores that allow substances to pass into and out of the nucleus. They're guarded by nuclear pore complex, which regulates the transport of specific large molecules. So not everything is allowed to enter the nucleus. The nucleus is well protected, and not everything is allowed to leave the nucleus. So for example, something like DNA would not be allowed to leave the nucleus. This figure shows really nice picture of the cell nucleus and its components. So it's got the nuclear envelope, which is which has nuclear pores, and nucleoplasm, which contains the chromatin, and the nucleolus at the center. So now we're at the nucleoli. Nucleolus is one, nucleoli is plural. So that's these dark staining round bodies within the nucleus that are involved in ribosomal RNA synthesis and ribosome subunit assembly. So the ribosomes are made at the level, these subunits, remember there's a large subunit and the smaller subunit is made at the level of the ribosomes, and then they're assembled. It's associated with the nuclear organizer regions that contain the DNA that codes for ribosomal RNA, and usually one or two per cell. The chromatin, which is found within the nucleoplasm, consists of 30% thread-like strands of DNA, 60% of histone proteins that are associated with that DNA, and 10% RNA. It's arranged in fundamental units called nucleosomes, which consist of DNA wrapped around those histone proteins. Chemical alterations of histones have an effect on DNA and therefore can help regulate gene expression. Chromosomes are, when we see chromosomes, they're basically condensed chromatin. So chromatin that has been wrapped really tightly. The condensed state helps protect fragile chromatin threads during cell division. This figure shows the structure of a chromosome. Okay, so if we start, chromosomes are made up of the double helix, and it's wrapped around these beads of nucleosomes, and to form, so it's wrapped around twice, in fact. Every DNA strand is wrapped around twice, and so the whole thing is called the nucleosome, and then that forms the chromatin fiber, and once it's really coiled and packed, it's a chromosome. 3.26a, chromatin and chromosome structure. Okay, so here again, if you start at 1, DNA is the double helix, and it's wrapped around those histones. It's wrapped around twice. In fact, around 8 of those. So nucleosome is made up of, it's about 10 nanometers in diameter. It's made up of 8 histone proteins wrapped by two winds of the DNA double helix. Figure 3.26b. So at this level of packing, you see a tight helical fiber. And then when the chromosome is at the midpoint of cell division, called metaphase, then it's made up of these two sister chromatids that are tightly packed together into this recognizable chromosome. So basically, on the left, you can see the nucleus containing the chromatin. So when so chromatin appears diffuse, it looks like a plate of spaghetti, okay? When the cell is non-dividing, when the cell is not cell does not have to divide, that's what it looks like. But as soon as the cell divides, then the chromatin is going to condense into that condensed chromosome that we see on the right. And now it's ready for cell division.
And this is also an animal. An animal carcass, animal carcass, again, animal. The thing that all of these other things have in common is that they're made out of the same basic building block, the animal cell. Animals are made up of your run-of-the-mill eukaryotic cells, and these are called eukaryotic because they have a true kernel in the Greek, a good nucleus, and that contains the DNA and calls the shots for the rest of the cell, also containing a bunch of organelles. There's a bunch of different kinds of organelles, and they all have very specific functions, and all of this is surrounded by the cell membrane. Of course, plants are eukaryotic cells too, but they're set up a little bit differently. Of course, they have organelles that allow them to make their own food, which is super nice. We don't have those. And also, their cell membrane is actually a cell wall. It's made of cellulose. It's rigid, which is why plants can't dance. If you want to know all about plant cells, we did a whole video on it. And you could click on it here if it's online yet. It might not be. A lot of the stuff in this video is going to apply to all eukaryotic cells, which includes plants and fungi and protists. Now, rigid cell walls, that's cool and all, but one of the reasons that animals have been so successful is that their flexible membrane, in addition to allowing them the ability to dance, gives animals the flexibility to create a bunch of different cell types and organ types and tissue types that could never be possible in a plant. The cell walls that protect plants and give them structure prevent them from evolving complicated nerve structures and muscle cells that allow animals to be such a powerful force for, eating plants. Animals can move around, find shelter and food, find things to mate with, all that good stuff. In fact, the ability to move oneself around using specialized muscle tissue has been 100% trademarked by Kingdom Animalia.
What about protozoans?
Excellent point. What about protozoans? They don't have specialized muscle tissue. They move around with cilia and flagella and that kind of thing. So way back in 1665, British scientist Robert Hooke discovered cells with his kind of crude beta version microscope. He called them cells because they looked like bare Spartan monks' bedrooms with not much going on inside. Hooke was a smart guy and everything, but he could not have been more wrong about what was going on inside of a cell. There is a whole lot going on inside of a eukaryotic cell. It's more like a city than a monk's cell. In fact, let's go with that. A cell is like a city. It has defined geographical limits, a ruling government, power plants, roads, waste treatment plants, a police force, industry, all the things a booming metropolis needs to run smoothly. But this city does not have one of those hippie governments where everybody votes on stuff and talks things out at town hall meetings and crap like that. Nope. Think fascist Italy circa 1938. Think Kim Jong-un's North Korea, and you might be getting a closer idea of how eukaryotic cells do their business. Let's start out with city limits. So as you approach the city of Eukaryopolis, there's a chance that you will notice something that a traditional city never has, which is either cilia or flagella. Some eukaryotic cells have either one or the other of these structures, cilia being a bunch of tiny little arms that wiggle around and flagella being one long whip-like tail. Some cells have neither. Sperm cells, for instance, have flagella and our lungs, and throat cells have cilia that push mucus up and out of our lungs. Cilia and flagella are made out of long protein fibers called microtubules, and they both have the same basic structure. 9 pairs of microtubules forming a ring around 2 central microtubules. This is often called the 9+2 structure. Anyway, that's just so you know when you're approaching the city, watch out for the cilia and flagella. If you make it past the cilia, you will encounter what is called a cell membrane, which is a kind of squishy, not rigid plant cell wall, which totally encloses the city and all of its contents. It's also in charge of monitoring what comes in and out of the cell, kind of like the fascist border police. The cell membrane has selective permeability, meaning that it can choose what molecules come in and out of the cells for the most part. And I did an entire video on this, which you can check out right here. Now, the landscape of Eukaryopolis, important to note, is kind of wet and squishy. A bit of a swampland. Each eukaryotic cell is filled with a solution of water and nutrients called cytoplasm. And inside of this cytoplasm is a scaffolding called the cytoskeleton. It's basically just a bunch of protein strands that reinforce the cell. Centrosomes are a special part of this reinforcement. They assemble long microtubules out of proteins that act like steel girders that hold all the city's buildings together. The cytoplasm provides the infrastructure necessary for all the organelles to do all of their awesome, amazing business, with the notable exception of the nucleus, which has its own kind of cytoplasm called the nucleoplasm, which is a more luxurious, premium environment befitting the cell's beloved leader. But we'll get to that in a minute. First, let's talk about the cell's highway system. The endoplasmic reticulum, or just ER, are organelles that create a network of membranes that carry stuff around the cell. These membranes are phospholipid bilayers, same as in the cell membrane. There are two types of ER. There's the rough and the smooth. Fairly similar, but slightly different shapes, slightly different functions. The rough ER looks all bumpy because it has ribosomes attached to it, and the smooth ER doesn't, so it's a smooth network of tubes. Smooth ER acts as a kind of factory warehouse in the cell city. It contains enzymes that help with the creation of important lipids, which you'll recall from our talk about biological molecules. Phospholipids and steroids that turn out to be sex hormones. Other enzymes in the smooth ER specialize in detoxifying substance, like noxious stuff derived from drugs and alcohol, which they do by adding a carboxyl group to them, making them soluble in water. Finally, the smooth ER also stores ions in solutions that the cell may need later on, especially sodium ions, which are used for energy in muscle cells. So the smooth ER helps make lipids, while the rough ER helps in the synthesis and packaging of proteins. And those proteins are created by another type of organelle, the ribosome. Ribosomes can float freely throughout the cytoplasm or be attached to the nuclear envelope, which is where they're spat out from. And their job is to assemble amino acids into polypeptides. As the ribosome builds an amino acid chain, the chain is pushed into the ER. When the protein chain is complete, the ER pinches it off and sends it to the Golgi apparatus. And the city that is the cell, the Golgi, is the post office, processing proteins and packaging them up before sending them wherever they need to go. Calling it an apparatus makes it sound like a bit of complicated machinery, which it kind of is, because it's made up of like these stacks of membranous layers that are sometimes called Golgi bodies. The Golgi bodies can cut up large proteins into smaller hormones and can combine proteins with carbohydrates to make various molecules, like for instance, snot. The bodies package these little goodies into sacs called vesicles, which have phospholipid walls just like the main cell membrane, then ship them out either to other parts of the cell or outside the cell wall. We learn more about how vesicles do this in the next episode of Crash Course.
The Golgi bodies also put the finishing touches on the lysosomes. Lysosomes are basically the waste treatment plants and recycling centers of the city. These organelles are basically sacs full of enzymes that break down cellular waste and debris from outside of the cell and turn it into simple compounds which are transferred into the cytoplasm as new cell-building materials.
Now, finally, let us talk about the nucleus, the beloved leader. The nucleus is a highly specialized organelle that lives in its own double-membraned, high-security compound with its buddy, the nucleolus. And within the cell, the nucleus is in charge in a major way. Because it stores the cell's DNA, it has all the information the cell needs to do its job. So the nucleus makes all the laws for the city and orders all the other organelles around, telling them how and when to grow, what to metabolize, what proteins to synthesize, how and when to divide. The nucleus does all this by using the information blueprinted in its DNA to build proteins that will facilitate a specific job getting done. For instance, on January 1st, 2012, let's say a liver cell needs to help break down an entire bottle of champagne. The nucleus in that liver cell would start telling the cell to make alcohol dehydrogenase, which is the enzyme that makes alcohol not alcohol anymore. This protein synthesis business is complicated. Lucky for you, we will have, or may already have, an entire video about how it happens. The nucleus holds its precious DNA along with some proteins in a web-like substance called a chromatin. When it comes time for the cell to split, the chromatin gathers into rod-shaped chromosomes, each of which holds DNA molecules. Different species of animals have different numbers of chromosomes. We humans have 46, fruit flies have 8. Hedgehogs, which are adorable, but less complex than humans, have 90. Now the nucleolus, which lives inside of the nucleus, is the only organelle not enveloped by its own membrane. It's just a gooey splotch of stuff within the nucleus. Its main job is creating ribosomal RNA, or rRNA, which it then combines with some proteins to form the basic units of ribosomes. Once these units are done, the nucleolus spits them out of the nuclear envelope where they are fully assembled into ribosomes. The nucleus then sends orders in the form of messenger RNA, or mRNA, to those ribosomes, which are the henchmen that carry out the orders in the rest of the cell. How exactly the ribosomes do this is immensely complex and awesome. So awesome, in fact, that we're going to give it the full crash course treatment, an entire episode. And now for what is, totally objectively speaking, of course, the coolest part of the animal cell. Its power plants, the mitochondria, the smooth, oblong organelles where the amazing and super important process of respiration takes place. This is where energy is derived from carbohydrates, fats, and other fuels and is converted into adenosine triphosphate, or ATP, which is like the main currency that drives life in Eukaryopolis. You can learn more about ATP and respiration in an episode that we did on that. Now, of course, some cells, like muscle cells or neuron cells, need a lot more power than the average cell in the body, and so those cells have a lot more mitochondria per cell. But maybe the coolest thing about mitochondria is that long ago, animal cells didn't have them, but they existed as their own sort of bacterial cell. And one day, one of these things ended up inside of an animal cell, probably because the animal cell was trying to eat it. But instead of eating it, it realized that this thing was really super smart and good at turning food into energy, and it just kept it. It stayed around. And to this day, they sort of act like their own separate organisms, like they do their own thing within the cell, they replicate themselves, they even contain a small amount of DNA. Now, what may be even more awesome, if that's possible, is that mitochondria are in the egg cell when an egg gets fertilized. And those mitochondria have DNA. But because mitochondria replicate themselves in a separate fashion, it doesn't get mixed with the DNA of the father. It's just the mother's mitochondrial DNA. That means that your and my mitochondrial DNA is exactly the same as the mitochondrial DNA of our mothers. And because this special DNA is isolated in this way, scientists can actually track back and back and back and back to a single mitochondrial Eve who lived about 200,000 years ago in Africa. All of that complication and mystery and beauty in one of the cells of your body. It's complicated, yes, but worth understanding. Review time! Another somewhat complicated episode of Crash Course Biology. If you want to go back and watch any of the stuff that we talked about it to reinforce it in your brain, if you didn't quite get it, just click on the links and it'll take you back in time to when I was talking about that mere minutes ago. Thank you for watching. If you have questions for us, please ask below in the comments or on Twitter or on Facebook, and we will do our best to make things more clear for you. We'll see you next time.